And I teach here at Villanova the Great Book Seminar, which sometimes includes Darwin and also philosophy. One of my interests in philosophy is philosophy of biology, so it's my great honor, especially honored, to introduce Dr. Evelyn Fox Keller, who's one of the most distinguished historians and philosophers of biology. Dr. Keller took her PhD from Harvard University in physics. She presently teaches at MIT. She specializes, as I said, in history and philosophy of biology, and also questions of gender and science, and the connections, deep and perhaps sometimes surprising, between theoretical biology and issues in gender. She's the author of many highly and rightly praised books. Two of those books bear special relevance to today's topic, and those books are Refiguring Life, Metaphors of 20th Century Biology, and The Century of the Gene, and I've learned a lot from both those books. They're really excellent. To give you just one provocative quotation from The Century of the Gene, and I think we'll hear some arguments along these lines in her talk, it seems evident that the primacy of the gene as the core explanatory concept of biological structure and function is more a feature of the 20th century than it will be of the 21st century. So with that, please join me in welcoming Dr. Evelyn Fox Keller.
Well, thanks very much for the introduction, for the invitation. Thank you for coming. I wish I had a comparable skill. I must say, it was a very hard act to follow. It was a tour de force. Okay, so I begin with a quote that you all know. And what I want to do, I should say, is give a very brief and caricature history of particulate units of inheritance, and I will do so by hitting on some well-known quotations to mark certain basic fundamental beliefs and arguments. And one of these quotations is Wilhelm Johannsen's, when he introduced the term, when he coined the term gene, and he wrote, "The word gene is completely free from any hypotheses. It expresses only the evident fact that, in any case, many characteristics of the organism are specified in the gametes by means of special conditions, foundations, and determiners which are present in unique, separate, and thereby independent ways. In short, precisely what we wish to call genes." And 2 years later, Johannsen added, "The gene," he says, because obviously the question is, what is this gene? And well, it's nothing but a very applicable little word, easily combined with others, and hence it may be useful as an expression for the unit factors, the elements, or allelomorphs in the gametes, demonstrated by modern Mendelian researches. As to the nature of the genes, it is as yet of no value to propose any hypothesis, but that the notion of the gene covers a reality is evident in Mendelism. Now, what was this reality that was so evident in Mendelism? I claim that that reality lay in the properties the gene was intended to account for. For Mendel, it was the sorting ratios, but for other people, it was more general characteristics, properties that preceded, I might say, both temporally and logically, Mendel's findings in his experiments on plant hybridization. Long before the rediscovery of Mendel's work, long before Johannsen's coining of the word gene, biologists have been looking to particular units of inheritance to ground these properties. Mendel himself called these units elementi. Darwin called them gemmules, Galton stirps, Weismann determinants, De Vries pangens. Now, I'm interested in the move, even independent of Mendel's findings, the move to look for particulate, discrete, separable units. And I have argued that a dominant influence, a dominant push to that formulation was the success of atomic theory and chemistry in the 19th century. that where there had been an enormous success in chemistry to the notion, enormous validation of the notion that you could ground generic properties in atomic units. So one of the properties, I might say, one of the properties that needed to be accounted for by genes or the other particulate precursors were hypothesized that the gene, one of the properties that these particles were hypothesized to account for was the stability of the phenotype, its persistence through the generations. So, for example, here's Weissman on the subject. When we find in all species of plants and animals 1,000 characteristic peculiarities of structure continue unchanged through long series of generations, when we even see them in many cases unchanged throughout whole geological periods, we very naturally ask for the causes of such a striking phenomenon. How is it that a single cell can reproduce the tout ensemble of the parent with all the faithfulness of a portrait? Indeed, half a century later, with genetics well into its stride, this is precisely the property that led Erwin Schrödinger, the physicist, to discover, it just drew him to the gene. And where Schrödinger wrote, speaking of portraits, following directly from Weissman, looking at the portraits of the Habsburg dynasty, he notes that several members of that dynasty have a peculiar disfigurement of the lower lip, the Habsburger Lippe. Fixing our attention on the portraits of a member of the family in the 16th century and of his descendant living in the 19th, we may safely assume that the material gene structure, this thing, responsible for the abnormal feature, has been carried on from generation to generation through the centuries, faithfully reproduced at every one of the not very numerous cell divisions that lie between. Again, the question, how are we to understand that it has remained unperturbed by the disordering tendency of the heat motion for centuries? How are we to understand that permanence, that extraordinary stability of the thing that underlies the trait that we see? But the gene, as all its particulate precursors, particulate precursors, was meant to do much more. Responsible not only for the extraordinary stability of phenotypic memory, it also had to be able to direct and control the processes of development in every individual organism. Despite the difficulty of imagining what kind of an entity might do this, what kind of an entity might both have the rock solidity to guarantee the permanence, the fidelity of the Habsburger Lippe through centuries, and the creativity to direct the unfolding of the organism, despite the difficulty of imagining what kind of an entity that might be, the confidence that some such unit existed was really remarkable, that there had to be a thing that would do both of these jobs. And as I said already, I am convinced that that confidence drew enormous strength from the successes of the atomic theory of matter. And in fact, de Vries himself makes such a reference. He wrote, "Just as physics and chemistry are based on molecules and atoms, even so, the biological sciences must penetrate to these units in order to explain by their combinations the phenomena of the living world." References, de Vries was not the only one. There are scattered references throughout the literature to the successes of physics and chemistry. And the parallels with biology were frequent in the latter part of the 19th century, and they continued well into the 20th century. So here is H.J. Muller in 1916, who wrote about the curious similarity which exists between the main problems of physics and of biology. Now, Muller is writing in the age, in 1921, he's writing in the age of the early quantum mechanics. And so he's not only relying on the atomic theory of chemistry, but he is developing it further because he draws from physics a new inspiration from, where he writes, it's not physics alone which has its quantum theory. Biological evolution too has its quanta. These are the individual mutations. And a very famous paper of his entitled Artificial Transmutation of the Gene, which is a direct copy of the artificial transmutation of the atom that had appeared not too long before. He wrote, mutation and transmutation are the two keystones of our rainbow bridges to power. Now, this hypothesized entity called a gene, later called a gene, also received an enormous boost from microscopy. Because the ability to see chromatin fibers made these fibers, the very fact that you could see them through the microscope, made these elements, the chromosomes themselves, concrete. And then later, when these elements could be tied to inheritance, that seemed to provide enormous support to the idea that there were things in the cell. You could see them. You couldn't touch them yet, but you could see them. They must be these particles that underlie inheritance. In retrospect, however, we are much more sensitive to how misleading that kind of microscopic evidence, in fact, was. I mean, in order to see the chromosomes, you couldn't see the chromosomes unless you used stains developed in the 19th century, which killed the cell. And completely erased entities that were dynamic or could not absorb the stain. But there was the evidence. It was there. It was concrete. Okay. So the question then becomes, for the 20th century, what are these elementary units of life? What are the genes out of which we are going to build a biology? As De Vries himself wrote, they're not the chemical molecules. They're much larger than these and are more correctly to be compared with the smallest known organisms. In fact, that was how De Vries resolved, in a way, reconciled the dual properties, this duality of the function of the gene, its permanence, its ability to guarantee the permanence and also to direct the development. And another quote, I don't know if this is on a slide, no. Yes, it's on Thomas Mann. Another quote. Thomas Mann is always a great source for. Thomas Mann was a writer who was enormously interested in current biological writings, an avid follower, and thought rather carefully, had a number of is a wonderful source for a writer like me to quote, because he just captured exactly the sort of essence of so many issues. And as he wrote, as long as one spoke of living units, one could not correctly speak of elementary units, for there was no such thing as elementary life, in the sense of something that was already life and yet elementary. And the gene was exactly something like this. It was, at least for De Vries, it was both, it had to be elementary and already alive, the smallest possible organism. 1911, Alvin Hagedorn, who wrote, to try to explain the facts of evolution and inheritance by the behavior of living particles, which have been invented simply to admit of this explanation, he wrote, that's an excerpt from a sentence to call attention to the paradox of the paradoxical nature of the very adventure. And later, Muller himself, no mean critic of his contemporaries as well as of himself, those who postulated, and his precursors, those who postulated pangenes, determinants, or other self-reproducing particles in the old days did not seem to realize what a monster they had by the tail. They were still subconsciously so close to the ancient lore of animism, in which practically all things were living, that to attribute reproduction to a particle itself living anyway seemed to present no problem. Nevertheless, this duality persisted through, I would say, it persisted in the form of the particulate unit under the name of the gene. I would argue it persisted through to the middle of the 20th century, at least till 1953. And it was the duality between what the gene does and what it is. And in many ways, its persistence was guaranteed by the fact that the dualism, the duality of the concept of the gene, and I've called it, it was simultaneously atom and organism, that duality was hidden out of sight, and enabling that notion, that idea of the gene to allow research to continue, to allow research to go on, to be enormously productive, and occluding, keeping out of sight the fundamental problem that there was no means to address. And in fact, what I've argued in the Century of the Gene, which was just mentioned, I claim a crucial metaphor, a crucial concept that worked to enable both to enable research to go forward and to occlude, to keep out of sight the hidden problems, was the concept of gene action. Gene action was, as Sturtevant and many other people argued repeatedly, gene action was what took you from the gene to the phenotype, or from the genotype to the organism. Sturtevant mapped out, he defined the aim of genetics, the ability of genetics to solve the problem of embryogenesis, and as a project to identify the activity of all of the genes, to trace their, what did he call them? To trace their modes of action, the chains of action from the gene itself to the trait, or and by implication, from the whole genome to the organism. In fact, here's a quote. I don't think I have it on a slide. Yes, I do. He wrote, "One of the central problems of biology is that of differentiation. How does an egg develop into a complex, many-celled organism? That is, of course, the traditional major problem of embryology, but it also appears in genetics in the form of the question, how do genes produce their effect?" And he wrote, "In most cases, there is a chain of reaction between the direct activity of a gene and the end product that the geneticist deals with as a character. Genetic experiments can therefore approach the problem through the analysis of certain chains of reactions into their individual links." I've called, in a number of things I've written, I've referred to this, the use of the concept of the metaphor of gene action, and it's associated, what all of it implied, is the discourse of gene action, a discourse that was particularly dominant in American genetics. And it enforced a particular, as I say, it was very productive, it was in allowing research to go on, but it enforced a particular representation of development, a spatial and temporal representation that precluded other ways of looking at development. The temporal representation was one that begins with, when does the gene begin to act? At the moment of fertilization. So development begins, T equals 0 is fertilization. As the clock goes on, as the clock moves on, you move from fertilization to adulthood. Anything that happens before fertilization is either erased off a map, or it's moved, in the case of maternal effects, was moved to the end of life and relabeled late effects. And similarly, there was a spatial, I think I have this is not not coming through. Okay, Oscar Schotte, the embryologist, had a diagram that he'd like to draw that illustrated the spatial representation of the discourse of gene action, in which the fertilized cell is almost entirely filled by the nucleus. He says that's how the geneticist sees the fertilized cell, and drew another fertilized egg with the nucleus very small, and the rest filled with cytoplasm. That's how the embryologist sees it. So 2 different spatial and temporal representations. And von Hecker and many others made it very clear that the first representation, the beginning of life, beginning of development at t equals 0 with fertilization, and the cell as the fertilized cell as primarily the nucleus, was a representation that was typically that belonged to the American genetics community, and argued that in Germany they see things rather differently, but their perceptions, their contributions have been thrown out. Now, I've argued that, I don't want to go into this argument here, but I've argued that these metaphors, these representations, had really quite notable, you can track the influence it had on the kinds of questions that were asked, on the ways questions were asked, and even on the kinds of research efforts that were seen to be legitimate and that were encouraged. However, early molecular biology seemed to provide a total vindication for these, I'll call them 19th century dreams, 19th, early 20th century dreams. Early molecular biology taught us what a gene is, and appeared to vindicate all the hopes of classical genetics. The gene now, as a defined stretch of a double-stranded helix, is automatically provided with immortality by view of the replication process. That the stability of the gene is guaranteed simply by the fidelity of that replication, automatic fidelity of that replication process. And the central dogma provides an answer to what genes do. What does a gene do? Gene makes a protein. And it gave rise to a reformulation. This also doesn't come through, I'm sorry to say. A reformulation, but only a slight reformulation of the Weissmann diagram of development, in which a crude version is you have the germ line is the immortal germ line is presented by a set of arrows that goes from the replication of each germ line. In each generation, the germ line gives rise to the soma, but the soma does not perpetuate. In the central dogma, we have, thanks to Maynard Smith, we have exactly the same diagram, except it's the DNA that is perpetuated along this line that's trained to immortality, and the proteins replace the soma. Now, of course, the findings of early molecular biology were enormously impressive. So impressive that they were powerful, and they're so powerful that they, interestingly, although they vindicated so many of the expectations of classical genetics, they did force a change in language. For example, once we could say, or we thought we knew what a gene is, that discrete stretch of DNA sequence, the productive ambiguity that was in the concept of the gene prior to that its ability to simultaneously ground the immortality, the permanence, the stability of the phenotype, and to direct development in each generation, that ambiguity couldn't be sustained. The gene, it's true, it was not an atom, but it was a molecular, well-defined, discrete molecular segment. It was a defined part of a molecule, but more problematically, it was abundantly evident that the gene, this stretch of a nucleotide sequence, was not an organism. Perhaps it can make proteins, but even Francis Crick would have had to admit that between an organism and the superproteins lies an enormous gap. The synthesis of proteins had to be regulated, and the question was, how was this to be done? You could not really, in other words, knowing the chemical constitution of the gene precluded the hope that that itself would provide the kind of agent of design that development mandated. Monod and Jacob introduced two very felicitous notions that helped enormously. One was regulator genes, and the other was the genetic program. I won't talk about regulator genes here, but just a word about a genetic program. What is a genetic program? Well, just like the classical gene, this too no one could say. But crucially, like the classical gene, it filled the gap. I suggest that the fundamental ambiguity of the genetic program was a crucial invention, was a crucial ingredient for maintaining the momentum of research in the early days of molecular biology. But erosion of this vindication of classical genetics started early, and particularly erosion of the idea that you could understand the permanence, genetic immortality, just from the structure of the DNA itself. And certainly by the 1980s, the simple idea of stability as built into the structure of DNA was pretty much done in. There is, in fact, nothing automatic about the fidelity of DNA replication. Rather, as we now know, the astonishing stability of genomic sequences and the replication of genomic sequences, which is certainly real, is the effect not of the structure of DNA, not of the double helical or complementary pairing, but rather it's the effect of an exceedingly complex and dynamical system of editing and repair that involves an enormous number of other molecules and very complex molecular machines. Similarly, the simple answer early molecular biology had offered to what a gene does, namely that it makes a protein, with its implicit one gene, one enzyme hypothesis, has also eroded quite a bit, before the end of the 20th century, probably most precipitously by the discovery of alternative splicing and the recognition that many genes, often in many cases thousands of proteins, could be produced by a single sequence of DNA. Indeed, it was this simple vision of classical and early molecular genetics that underlay the launching of the Human Genome Project. The realization of that program, however, beginning with its surprising report that the human genome has only 25 to 30,000 genes, has ever since that early 2000, has systematically contributed to the subversion of the simple, I might say, elegant dream that had motivated it. Now, in 2000, the population geneticist Jerry Coyne reviewed my book rather unkindly, and he wrote, "The century of the gene is in fact a jihad against our notion of the gene." To Keller, gene is simply an outmoded term, a semantic straitjacket signifying something that can't be defined. But the notion that geneticists are semantically challenged is simply silly. You don't laugh, but you're allowed to laugh, because today the notion that the gene may be an outmoded term and that as geneticists we might say we are semantically challenged is everywhere. For example, just a couple of years ago we read in Nature, here's another slide, where the meaning of most four-letter words, is all too clear, that of gene is not. The more expert scientists become in molecular genetics, the less easy it is to be sure about what, if anything, a gene actually is. Here, for example, is the result of a lengthy and very contentious 2-day-long attempt of a group of information biologists to come up with a definition that people could agree upon. Here it is. A gene, very simple, right? A gene, just a simple word. A gene is a locatable region of genomic sequence corresponding to a unit of inheritance, which is associated with regulatory regions, transcribed regions, and or other functional sequence regions. Got it? Okay. Helen Pearson, who is the author of the first quote that I gave you, goes on went on and wrote, instead of discrete genes dutifully mass-producing identical RNA transcripts, a teeming mass of transcription converts many segments of the genome into multiple RNA ribbons of differing lengths. These ribbons can be generated from both strands of DNA rather than from just one, as was conventionally thought. Some of these transcripts come from regions of DNA previously identified as holding protein coding genes, but many do not. Furthermore, many scientists are now starting to think that the descriptions of proteins encoded in DNA knows no borders, that each sequence reaches into the next and beyond. I should add that this is 2006. I should add another, just hold that a second. Since 2006, the whole revolution of RNA regulation it started before 2006. So genes are Helen Pearson's discussion of the enormous flexibility of the notion of gene is still focused on the coding of proteins, but now the concept of genes have been extended to people who even talk about the coding of RNA, whatever that might mean. And not only coding of proteins, not only transcription of RNA, but also the concept of the gene is extended beyond the DNA. So Susan Lindquist writes that most people think of genetics as being only about DNA, but genetics is about the inheritance of traits. While most traits are inherited through transmission of DNA, the traits we study are inherited through proteins. Thus, these proteins can be every bit as much a genetic element as DNA. After all, they are heritable. Just thinking about DNA sequences to see the magnitude of the problem. And with this typical acerbity, here's how Sidney Brenner sums up the situation in which we find ourselves. Old geneticists knew what they were talking about when they used the term gene, but it seems now to have become corrupted by modern genomics to mean any piece of expressed sequence. So, my question is, well, I thought there was more. Okay. So does this mean there's a crisis in genetic determinism? Well, yes. Certainly, one might conclude that such a crisis exists from the kinds of statements that have been used to justify the development of a new kind of biology. The human genome, that's systems biology, or the Human Genome Project itself might be regarded as the apotheosis of genetic reductionism. But in giving a list of parts, it revealed the inadequacy of such a list. In fact, genomics today has also begun to reveal the inadequacy of the very notion of a genetic part. In an illuminating recent review, Kopernoff, Willingham, and Gingras survey a large amount of evidence pointing to the unexpectedly complex ways in which DNA works. These all go well beyond the notion of a gene as a piece of DNA whose function is to produce a protein. They include the use of common pieces of DNA for multiple, often quite different purposes, the use of overlapping sequences in the same or in different functions, the importance of long-range elements in helping to bring about a function, head-to-head transcription on the one hand and faraway transcription brought together on the other, both sense and anti-sense transcription, both cis and trans mechanisms, and issues related to timing in the process of transcription and translation. All of these can be present or many of these can be present simultaneously in the process of going from genotype to phenotype. As Kapranoff and his colleagues write, these observations suggest that genomic architecture is not collinear, but is instead interleaved and modular, and that the same genomic sequences are multifunctional. Their argument has now been strikingly confirmed by the findings of the ENCODE Project Consortium. These findings are of enormous interest, and they dramatically underscore the need for new ways of thinking about DNA sequences, genomic organization, and their relationship to function. If we have learned nothing else, it is that relation is vastly more complex than we had ever anticipated. In a more recent paper, Gingeris, I don't know how to pronounce his name, Gingeris, suggests that this increased complexity necessitates a reconsideration of the definition of a gene and requires the use of an alternative term to help to define the fundamental operational unit that relates genomic sequences to phenotypes. As a crisis. Rather, I see it as an opportunity. There may not be any particulate units of inheritance. Indeed, it seems there isn't anything that can legitimately be called a genetic program either. But there certainly are molecules interacting in extraordinarily complex dynamical ways. All sorts of molecules, some more stable than others, lipids, sugars, metabolites, etc., and many of which have been crucially implicated in regulatory dynamics. None of this is to deny that DNA remains an amazing and extraordinarily versatile molecule, or that its role in the cellular economy is not to be seconded. But the powerful functions associated with this molecule are products of the ways in which it interacts with other molecules. It takes a cell to realize the many possibilities that lie latent in the DNA. And maybe, as some have suggested, DNA can be thought of as a kind of musical score or archive. But like a score, its function is potential, waiting for an orchestra and a conductor to interpret it. I'm certainly not saying, indeed I could not say, that the notion of the gene of a clear-cut, definable, and countable material entity has been given up, the idea of a parts list. If only for a lack of suitable alternative, we all continue to use the concept of the gene, I, myself included. But it has certainly begun to give way. On the one hand, references to this term are proliferating wildly. Not only, as Brenner puts it, any piece of expressed sequence, sometimes even unexpressed sequences, and for some, even heritable elements not composed of sequences, like prions. On the other hand, biologists are clearly beginning to reach for a different vocabulary, a vocabulary capable of doing greater justice to the dynamics of living systems. But we're still waiting for that vocabulary. We're still waiting for the Mendel of the 21st century. Thank you. Discussion, comments? Yes.
Well, thank you very much for that superb presentation. What do you think of the efforts of people like Craig Venter to create a synthetic biology, stitching together bits and pieces of genes to make both a chassis of elemental chassis into which one can put biobricks to perform particular biological functions?
Would you like a one-word answer?
I would, and then the expanded version.
Sure. My one-word answer is grossly, 2 words, grossly irresponsible. And then and then I'll expand. The irresponsibility. Okay, let me say, as I say, I do not. DNA is an amazing molecule. I repeat. It was probably the greatest innovation of evolutionary history, of biological evolution. DNA is also a locus of, it's a target for inducing transformations, biological transformations to an extraordinary extent. It does not need to be the only target of intervention for biological development to be a very powerful one. So to put together sequences, new kinds of sequences, is to introduce change. There's no question. It's to introduce change of a kind, of a nature that we are not able yet to predict. We do not have an understanding. We're developing a great deal of sophistication, but we are far from an understanding of the relationship between the ability to map genotype to phenotype. And part of the problem is that genotype to phenotype does not map univocally. So, nevertheless, the genotype is an enormous source of innovation. So what happens when we introduce new, unexplored, untested by evolution, new kinds of sequences? Nobody knows. What happens when we promote, as Drew Endy and company are doing, synthetic biology, synthetic biology, modular synthetic biology that can be easily circulated and transported, and not only are the units of DNA modularized, but the teaching is modularized too. So synthetic biology workshops are replicated and transported around the world. What happens? Well, it doesn't seem it doesn't seem a stretch to talk about, if we think about the proliferation of computer viruses, as computers became a backyard industry, to the proliferation of biological viruses as it becomes a backyard industry. I've asked Craig Venter about this, and he responds he can take care of it. He's aware of the problem, and he's keeping his eye on it. Well, that's good, but it's not very comforting. Yeah.
I know you're speaking from a historical perspective in the way you're doing. Do you think it is essential to posit a structural component for the gene, no matter what that is, in order to account for the stability we see in the phenotype?
Well, I'm not quite sure what you mean by the gene there, but let's talk about sequence, okay? Because we know what the structural components of sequence are, and we know that we cannot account for the stability of a sequence by its structure. We need lots of proteins that may may find their source in other sequences of the DNA. But the sequence itself will not. The structure of that sequence will not give you the whole story. Will the structure of the whole sequence, of the whole genome, give you the story? Again, the answer seems to be no. The answer seems to be no, because DNA is not it's not a static structure. It's a dynamic structure. It exists in multiple states, depending on the context, depending on the methylation, the histones binding to it. What can be done with DNA depends on its conformation, its methylation, etc. We can't separate out a single molecule and try to tell the story.